Good morning. I'm Dr. Neeraj Khatri Sapkota, and in this lecture, I'll be discussing on the concept of the blood in the course of human biology. So this concept is very clear to everyone that the blood is flowing all along the blood vessels, and through the blood vessels, this blood concept composition has to be learned. So when we draw the blood as a practical application, then we can just withdraw the blood from the arms, and then after withdrawing the blood we can put the blood in a special tube known as Winthrop tube. And when we put the blood in a Winthrop tube through the syringe, then we will then later place it on a machine known as centrifugation machine, and then we will revolve the blood that has been placed in the tube for about, let's say, 10 minutes or 15 minutes, and then we will see two layer conspicuously, one settling down and the another just floating all along the upper part of the tube. The one which has been floated is not heavier, and the one which are sedimented or present at the bottom is more heavier. So that means the heaviness of the lower part, that means this red part, is actually the red blood cells, and it contains about 45% of the total whole blood, whereas 55% of the whole blood is part of the plasma. So that means it is 55% of the whole blood is plasma, whereas 45% of the whole blood is red blood cells. The sedimentation or the settling of the red blood cell at the bottom is because of the presence of the hemoglobin, which contains iron, and its specific gravity now then become high. And what we saw is one layer is there that is known as buffy coat, which contains less than 1% of whole blood, that comprise of white blood cells and platelets. So these three layer are formed in a test tube or in the tube known as Winthrop tubes when the blood withdrawn from the arm is placed and then centrifuged for few minutes. So after seeing the blood, then we saw the red blood cells is the name given to that condition where the context, the theoretical value of this red blood cell is said to be hematocrit. So this hematocrit means it is the settlement of the red blood cells at the Winthrop tube. So this red blood cell along with other cells, for example white blood cells and platelets, together they are named as formed elements. So formed elements are said to be the red blood cells, white blood cells and platelets. The red blood cells, about 98% of the total formed element is formed by these red blood cells, whereas only 2% of the total formed element is formed by white blood cells. And these red blood cells, white blood cells has their own function, and that we will be studying in coming of lectures. But today we have understood the practical application of studying the way of the composition of the blood in the laboratory. So if we have such concept, then we can easily say that our blood contains plasma, formed element, plasma and formed element. Plasma contains electrolytes. Some proteins are even there, admixed with the fluid. Water is there, whereas the formed element are only said to have cellular element. So they are cellular element. They consist of cell. The true cell is said to be the WBC cells, whereas platelets and RBC cell, they do not have nucleus. RBC cell lost its nucleus during its formation, which are found by the hematopoietic stem cells or hematocytoblast. Whereas these WBC cells, they have clear conspicuous nucleus, and hence they are said to be the true cells. So with this concept of having the fluid and the cellular component of the blood, we would have more understanding of what does this component of the blood does in the body as a functional part. That will be studied in next lecture. For now, I'm going to close the session of the video for this section. So if you like my video, if you have taken some concept from my video, then I would request you to subscribe my video or give one like to my video, so that you can have vision, or you can see more other video when I posted.